So, dear students, today I am going to deliver a lecture on amoebiasis. So, amoebiasis is an infection with an intestinal parasite that is of protozoan origin, which is known as Entamoeba histolytica. It is particularly a tissue-lysing amoeba or tissue-lysing protozoan parasite. And this particular disease is mostly asymptomatic. Mostly 90% of patients having this particular disease doesn't show any symptoms. And particularly, this parasite can cause disease ranging from dysentery to different type of abscesses in the intestine, abscess in the liver, lungs, and brain occasionally. And mostly the asymptomatic infection is not due to Entamoeba histolytica by a different pathogen, a parasite that is known as Entamoeba disper. So both these parasites can cause more severe infections and also may cause fever, profuse diarrhea, abdominal pain, jaundice if they infect the liver, then anorexia, and eventually weight loss. In severe cases, it can lead to development of abscesses, means the pockets of amoeba as well as inflammatory cells. So they can create these abscesses in liver and very rarely in the brain. And why should we go for particularly to study this particular disease? Because our country, India, is in the endemic zone along with the other countries like Mexico and the tropical regions of Africa, South and Central America. So if you look back into the history of this particular disease is not a newly invented disease. So dates back in 1875, Loss differentiated amoebic dysentery from a bacterial dysentery that is actually known as bacillary dysentery caused by Shigella, caused by E. coli, enterotoxigenic or enteropathogenic E. coli. So Loss has identified that a particular organism that is not at all a bacterial origin is actually responsible for this dysentery. That is why he has termed that this is an amoebic dysentery and termed this pathogen as Amoebic coli. Later in 1891, Consilman and Lefleur, they have identified that ulcerative colitis that is also has a link with this amoebic dysentery and is particularly caused by a pathogen that is a protozoan origin. Then in 1902, Leonard Roger, who has published a research paper, citing 37 references, 37 cases of amoebic liver abscesses, AL disease, that has relation with Entamoeba histolytica. Later, next year, Fritz Sodin, he has established that this particular disease is caused by a protozoa, which is known as Entamoeba histolytica, is not an Amoeba coli or Entamoeba coli, it's not a bacteria. He has differentiated and established that this particular disease, dysentery or else a colitis, it is due to the infection of a protozoan parasite known as Entamoeba histolytica. Now we are coming into the epidemiology. So particularly among the protozoan disease or the parasitic diseases, amoebiasis is the third most common cause of death from parasitic diseases. Just fall after schistosomiasis or else malaria. So schistosomiasis and malaria, these are the two predominant diseases. After that, human that are responsible for a parasitic disease it is causing death in most common people is amoebiasis. And who are at the risk groups? The travelers, immigrants from the underdeveloping or developing countries, immunocompromised individuals, pregnant women, sexually active males, mostly who are the homosexuals, then the prisoners, children who are staying at the daycare centers, and the individuals who are the cis carriers. So how this particular disease is transmitted? This particular pathogen is transmitting from one human being to the another human being via fecal contamination of both the drinking water as well as the food. But also by direct contact with dirty hands and objects or objects as well as by sexual contact like homosexuals, you can also develop this particular disease. Additionally, geophagy means a common ritual there in some particular tribal communities of eating of earth or eating of soil. This may be a common route of infection in certain cultures. And people in developing countries that have poor sanitary conditions, immigrants that are coming from the developing countries, having same sanitary conditions, travelers to developing countries, then people who live in institutional having poor sanitary conditions like the daycare centers, prisons, as well as the immune compromised individuals like HIV positive patients and sexual transmission also are the modes of transmission of this particular parasite. Now I'm coming to the pathogenesis. So mostly the cyst, the mature cyst, they are resting coming when we are ingesting the mature cyst via contaminated water or food. It is coming and passing the stomach, acidic environment because this is resistant to acidic environment, and then resting at the small intestine or at the gut. After that, there you can see minute crypt lesions at the large intestine, and then extend through the uh, muscularis mucosa to the submucosa, where you can see this uh, see flask-shaped ulcer. Here you can see this is a picture flask-shaped ulcer at the submucosal layer, and then development of thrombosis of blood vessels as well as toxic megacolon. Here you can see deposition of cholesterol, and then blood circulation is stopped here in these blood vessels. So due to this thrombosis, so you can have this megacolon or enlargement of the colon due to inflammation, and then it will develop, eventually it will develop irreversible coagulation necrosis of the bowel wall. Here you can see this is the normal one, this is the normal colon, normal bowel, and this is the necrotic bowel. Here you can see necrosis because of non-availability of oxygen and food material. Here you can see this necrosis of this particular segment of your bowel, and they are developing this irregular coagulation necrosis of bowel wall. So this is the pathogenesis of this particular parasite. Now I'm coming into the life cycle. So life cycle starts with ingestion of the contaminated food or water. What we are injecting? We are actually eating, not injecting. We are actually eating the mature cyst or else the trophozoite. The mature cyst is present at the form stool, and the trophozoites are present at the diarrheal stool. If both the stools are contaminating either our drinking water or the food, and we are drinking it, we are ingesting as well the trophozoite and the mature cyst. Now the mature cyst will pass the stomach and then entering into the small intestine, where we can see excistation, means from the mature cyst the trophozoites are formed. The process is known as excistation. Then the trophozoites are coming, migrating to the from the small intestine to the large intestine, and they will undergo either multiplication by the process of a sexual mode of reproduction, that is binary fission, and later we will notice encystation. So encystation means from trophozoite again they will convert into cyst. So they will convert into cyst. The process is known as encystation, and they will produce the cyst, that is the immature cyst. Now here, when the trophozoite is there at the intestinal lumen, there may be three different phases, or three different types of infection. If the trophozoite is resting there at the intestinal lumen, confined at the intestinal, remain confined to the intestinal lumen, we can say this is non-invasive infection. But if the individual continuing to pass cyst in their stool, which are the asymptomatic carriers, the trophozoite can invade the intestinal mucosa. Then we can say this is intestinal disease, and if they reach the blood vessels or the and enriching the extra intestinal sites such as liver, lungs, or brain, occasionally, then we can say this is extra intestinal disease. So trophozoites multiply, as I said, trophozoites multiply by binary fission and produce cysts. And both stages are passed in the fishes. Cysts can survive days to weeks in the external environment and remain infectious in the environment due to the protection conferred by their walls. Now, the trophozoites passed in the stool are rapidly destroyed once outside the body, so they cannot stay for long. And if they ingested, would not survive exposure to the gastric environment. So it is only the cyst who can survive the gastric environment, not the trophozoite. And also the trophozoite cannot stay at the external body areas or else the external environment outside of the body. They cannot stay for long. So here, this immature cysts, they are multiplying not as a cell, only the nucleus multiplying from, and they will form a quadrinucleate structure that is known as a quadrinucleate cyst or multinucleate cyst. And this multinucleate cyst will exist from the host in the form of stool in case of non-invasive infection. So this is the detail. So ingestion of the cyst, excistation first, then conversion of trophozoite. The trophozoites will move and colonize in the colon, then encystation means formation of cyst. The cyst will come out, means excretion of the cyst, and then it will again ingestion by a patient or a new individual. So these are the two forms or two stages of Entamoeba histolytica that are actually responsible for transmission of this particular pathogen. One is the multinucleate cyst or the quadrinucleate cyst, and the second one is the motile trophozoite. But this one is a more dangerous one because they are resistant to the acidic environment of the stomach, and also they can survive outside the body for weeks, but not the trophozoite. They cannot even resistant to the acidic environment of the stomach. Now, Entamoeba histolytica pathogenesis, so it resists both the adaptive as well as the innate immunity of the host. The trophozoites first adhere with the colonic mucosal epithelial cell layers and leads to disruption. So, adherence of this particular cells at the mucosal epithelial cells is mediated by a family of surface selective molecule capable of binding to the galactose as well as the GalNAc, means N-acetyl galactosamine residues. So they can lyse host cells upon contact through a family of amphipathic peptides, which is known as amoebopores. So this picture will tell you. So first, they are binding here at the mucus layer, and then they are destabilizing it by using some by attaching with the lectin residues or the galactose and galanac binding sites or residues. Then they are invading to the submucosal layer by these pore forming proteins, which is known as amoebopore, and they are passing through these and then entering into the blood vessels, means metastasis via the portal vein, and it is happening only because of this amphipathic peptide, which is known as amoebopores. Now, this particular parasite passes a large family of possesses a large family of cysteine proteinases. So these cysteine proteinases is capable of lysing the extracellular matrix. Here, this is the extracellular matrix. So it is capable of lysing the extracellular matrix as well as combating with the complement system as well as the antibodies. Only because of these proteolytic antibodies, they can break the hinge region of the antibody into Fab and Fc regions. So now the antibody, they are not active. So they can cleaving the host defense molecules in the form of complement and antibodies. They are destroying this by using this particular cysteine proteinases. The ultimate effect of all this amoebic virulence factors on the human colon is the production of small ulcers that have hipped borders and contains focal areas of epithelial cell loss. And the trophozoites can then invade laterally through the submucosal layer and creating the classic class set ulcers that appear on pathologic examination as narrow necked lesions broadening the submucosal region. So Entamoeba histolytica found at the margin between the dead as well as the live tissues in the colon part. Now the symptoms are diarrhea, vomiting, abdominal pains, discomfort, and fever. And sometimes the symptoms it will take days, few days to weeks to develop and manifest themselves. And this particular disease is mostly dangerous in case of the immune compromised patient. Mostly they are asymptomatic, but this is dangerous for the immune compromised patient. Clinical findings, so you can find either the intestinal amoebiasis or else the extraintestinal lesions. So in case of intestinal amoebiasis, so asymptomatic infections, mild to moderate colitis. So mild colitis means non-dysenteric colitis to severe colitis like dysenteric colitis may happen. Localized ulcerative lesions of the colon you can notice, and also you can notice some localized granulomatous lesions in the colon, which is known as amoeba. So this amoeboma, which is the granulomatous lesions of the colon, this is happening only because of the infection of Entamoeba histolytica. But if the trophozoite invades and enters into the blood vessel, then we can see some extra-intestinal amoebiasis. And what are the organs? The organs are liver, lung, and brain. So we can only confirm by taking biopsy sample and then doing a cross section, tissue section, and then perform histopathological studies by hematoxylin staining, as well as GM staining will reveals the presence of trophozoites in the extra-intestinal lesions. So these are the sites where extra-intestinal lesions can happen, but brain it is very rare. So less than 1% cases. So invasive disease only 10%, mostly non-invasive. 90% is asymptomatic infection or non-invasive infection. Out of this 10%, less than 1% develop brain lesion via ingestion of Entamoeba histolytica cyst. So basically, an example of ALA, this amoebic liver abscesses. So in this particular disease, pus formation happen in liver abscesses, appears as a red uh, anchovy sauce-like appearances. The passes are this kind of appearances, this red anchovy sauce-like appearances, and the material aspirated is likely to contain trophozoites and may be detected by direct microscopic observations. So what are the basic methods for the diagnosis of this particular pathogen? First, you have to collect fresh stool. Then you have to go for wet mounting and permanently slide preparation and stain it using a trichome dye. So trichome dye will reveal these two type of pictures for the two type of strain. This one is Entamoeba histolytica and this one is Entamoeba dispar. Only the difference is here in the size of the nucleus, because both are almost similar in their morphology. So if you stain it using trichome dye, so it will stain it. The cell will stain and it will look like this. So via microscopic study you can identify this. But again, one more staining process is there by using Charcot Leyden crystals. This is again a staining process of stool examination to support the diagnosis of Entamoeba histolytica. So mostly the cysts are smooth and round set. They are devoid of glycogen mass. And stool sample possess numerous amount of RBCs and pus cells. These particular crystals, Charcot Leyden crystals, they are either diamond shaped or else the Wheatstone shaped. So here you can see the picture. So apparently they are colorless, but when you stain with this particular stain, they will stain purplish red via trichome. And vary in the shape. The shape maybe the size ranges from the length ranges from 50 more or less 50 micron, 40 to 50 micron in length. So in case of study whether it's an invasive one or non-invasive one, we have to go for serological test. So there are 4 different type of serological tests can be performed to confirm invasive amoebiasis. And the tests are indirect hemagglutination assay, enzyme immune assay, latex agglutination assay, and indirect immunofluorescence. So these are the tests you can confirm whether it's an invasive amoebiasis or it's a non-invasive amoebiasis. So culturing of bacteria is not routinely done. So a particular medium, that is Bock and Dolov lock egg medium, that has been modified by lead law and exclusively used for isolation and maintenance of Entamoeba histolytica. So to culture it, first you have to use a loop full of sterile rice powder. You have to add it with the medium mentioned here, and then you have to inoculate it a fresh feces or else a saline centrifugal sediment of the feces. You have to culture it 37 degrees centigrade for 2 days, and after 48 hours you have to subculture it 48-hour intervals, and then you can observe it under the microscope. So this is about the cultivation of this particular parasite Entamoeba histolytica. If you further confirm, because diagnosis of amoebiasis is very difficult by simple microscopic observation. So if you confirm if you go for further study, so best way is to isolate DNA from the feces or stool. Then you have to go for PCR, and this is the most sensitive and specific method for the identification. But only used by the for mostly for the research and epidemiological tool, not by the pathological centers, because it's very costly. And second one is ELISA technique using specific antigen of Entamoeba histolytica as well as Entamoeba dispar. This will clarify whether the disease is caused by histolytica or dispar. So these are the studies, advanced studies. You can use it for the diagnosis of Entamoeba histolytica as well as Entamoeba dispar. Now treatment. So mostly metronidazole is a compound that is the choice of drug. The to date Entamoeba histolytic has not demonstrated resistance to any of the compound of metronidazole or tinidazole. So if this does not work, so we have to administer chloroquine, emetine, and hydroemetine. So eliminating cysts in case of the carriers who do not have symptoms. So there we have to administer furamide, idoquinol, and uh, paromomycin. So paromomycin as well as or idoquinol is a preferred drug for ALA treatment as well as for amoebiatic colitis treatment. And then nitazoxanide can also be used as because it's a broad spectrum antiparasitic drug, so it has efficacy against Entamoeba histolytica trophozoites, both which is present in the tissue as well as in the gut. So you can use this particular compound, broad spectrum antiparasitic drug. Next is a prevention. So better hygiene is must to prevent this particular disease. So what you are drinking, you're drinking water. If it's a bottled water, then it's fine. If not, you have to at least boil it for 1 minute. 10 minutes is preferable. Boiling of water for 10 minutes is preferable. And never use any health drink, any orange juices or any other kind of juices that is prepared with ice cube. So it's not safe. And water can be made safe by filtering through the filtering, and the filter pore must be 1 micron or less. If it is not possible, by dissolving iodine tablet, filter water can, simple filtered water can also be taken as a drinking water. And you must avoid milk, cheese, dairy product that may not have been pasteurized. So always you have to eat pasteurized milk or cheese or other dairy product. Avoid anything sold by the street vendors because hygiene is not properly maintained there in most of the cases. Thoroughly cooked all raw foods and vegetables. And if you are using a pre-cooked food, reheat the food until it reaches the temperature at least 167 degrees Fahrenheit. Before preparation of food, before eating, before after going to toilet, changing diaper of a baby, after smoking, all the cases you have to wash your hand. And never share a tissue or a handkerchief or towel of others. So to maintain your personal hygiene, you have to use both soap and hot running water. And at least for 10 seconds, you have to rub your hands after toilet and changing a baby's diaper. And the second thing you have to do, clean your bathroom as well as toilet very often. And pay particular attention to the toilet seats and taps. Avoid sharing towels. So health education for improved water supply by using chlorination, by the chlorination process is mandatory. So the government should take precautions for that, and that's all about amoebiasis.